Hello everyone, I'm Rucha Gadre. I'm working in a IIT Bombay uh, SPR facility. So first of all, I would like to thank BioPractify team for this wonderful initiative and also like giving me opportunity to be a part of it. And a warm welcome to all the attendees. So okay, so today I'll be presenting a talk on a SPR, that is Surface Plasmon Resonance. So what we will be covering is introduction and assay development and a few applications of it. So the introduction. So let's begin with what is surface plasmon resonance. So it is a phenomenon that occurs where electron in a thin metal sheet becomes excited by a light that is directed to a sheet with a particular angle of incidence, and then it travels parallel to the sheet. So this phenomenon is designed is used to design an optical instrument which is used to study biomolecular interaction. So SPR phenomenon is used in a designing of a optical technique which is used to study biomolecular interactions. It is a label-free, SPR is a label-free technique. That means we don't need to tag our molecule. So we can use it in its native form. It is a real-time assay. That means we don't need to wait until the entire assay get over. We get the we get the information in a real-time manner. As the event occurs, we get the information. So what interaction studies we can study? We can study protein, protein nucleic acid, protein small molecule, antibody protein, protein lipid, etc. So first of all, why do we need to study a why do we need to study a biomolecular interaction? What is the use of it? So in biological system, there are several molecular interactions are taking place. For example, protein, protein DNAs with small molecule, enzyme substrate, etc. These interaction governs all sort of cellular processes like signal transduction, gene expression, metabolic pathways, etc. That's what become very crucial to understand how this system functions and get regulated. So this information will give us basic fundamentals of how the cellular or homeostasis is governed. Also, this information can be further explored to study, like since we know the basic fundamentals of this, uh, basic fundamental in basic fundamentals of our biological system. So this can be further studied. This can be further used in designing a drug molecule. We can engineer a molecule which can perturb. We can study a disease which can perturb this kind of interaction in disease progression or etc. If it is involved in a disease progression. So about SPR. So that's why it is very important to study this biomolecular interaction. There are different other methods are also available. For example, for qualitatively, we can use pull down assay, co-IP. For quantitative, we can use ITC. We can use, of course, this SPR. We can use BLI, etc. So now we'll focus on SPR. So SPR is an optical technique in which we study biomolecular interaction in a real-time manner, and we don't need to use any tag in this. So it is tag-free. So what is the principle of it? So SPR has a sensor chip which has a gold layer. It has I mean, we use interacting partner. So one of the interacting partner is immobilized and other is flown. So the partner which we immobilize is called as ligand, and the one which we flow is called as analyte in SPR. There is a light source and a detector. As we can see
Your
here,
slides are not changing.
slides are not seen. Okay.
Yeah,
Is it
now
visible?
they are.
Now there. Okay, sorry. So as you can see here, there is a light source which is incident on a chip. This chip has a gold layer, and there so this light monochromatic light is incident on a chip, and there is a total internal reflection. As the some binding occurs, there will be a change in this total internal reflection. This change is detected by the detector, and this change in angle is plotted against time, and this gives us information about the binding effect. This is the basic principle of SPR. Now we'll just see in a detail as we were discussing. This is a chip which has a metal film. The binding partner which we immobilize is called as ligand, and the one which is flown is called as an analyte. So light is a monochromatic light is incident on a metal film, and there is total internal reflection. Uh, and the detector will detect this. As a binding happens, the as the light is incident, there is an excitation of a surface plasmon result in a dark line in a reflected beam, and the angular position of dark line shift as the molecular binding event happens there. This angular shift versus time will give you the binding data. Now, so like till now, what we know that uh, like um, this SPR data is how we get the SPR data. So there is a ray of light incident on a metal film, and there is total internal reflection. This change in angle by time will give you the data. So what are the major events? First is association, and in association after immobilization, the analytes are flown. Analytes are flown and they bind. This phase is called as association phase. And as the after association, the buffer is flown and the analyte come off. This phase is called as dissociation. So these are two major events, that is association and dissociation. So if the analyte is strong enough, so in after flowing buffer it will not come off completely. So in that case we will need to regenerate our ligand. Regeneration means we will take out the analyte forcefully from the ligand without affecting the ligand, so that next round of analyte or next concentration of analyte can be passed. So this is how these are the major events, that is association phase, dissociation phase, and the regeneration. So this is how our typical sensogram looks. So this is association phase and this is dissociation phase. Now I'll just show you pictorially the machine which we have it here, that is Biaco T200. The chip is inserted here, buffers are kept in this bottle, and this is this is sample compartment. So this is the Biaco T200. We have it here in IIT Bombay. Now we will discuss about the assay development. There are three major steps in assay development. First is immobilization, other is sample injection, and third is regeneration, as we were discussing. So
Your
immobilization is
slide
the
is
process.
still the same.
Oh,
Now it's okay.
is it okay like this, or should I make it full screen?
Yes, ma'am.
Okay, should I make it full screen? Is it fine now?
Yeah, it would be better.
Okay. So the first step is immobilization. Immobilization, in which we immobilize one of the interacting partner, that is we called as ligand. Okay, we immobilize the ligand on the sensor chip. And then the analyte is flown. Okay, and we get the sensogram, and then we do the regeneration process. So the first step is surface preparation, that is for immobilization. Now, so immobilization is of two types, that is direct and the indirect. So in direct immobilization method, a ligand is linked to the surface directly. Okay, is linked to the surface directly. We can explore different chemistries for that. For example, amine, like ligand, thiol, surface thiol, aldehyde, etc. Hello, hello.
Somebody was unmuted. No, you
I
can
got
continue.
unmuted. Should I? Okay, okay, fine. So is the slide is visible now?
Yes, ma'am.
Okay. So in direct method, in direct method we directly immobilize ligand on the surface of the chip. It is covalent immobilization. So we can use different chemistries for that. For example, amine coupling, ligand, thiol, surface thiol, aldehyde, etc. Other method is capture method, in which we capture a molecule on the surface of the chip. Here it is an indirect method. For example, there are different chips available for this. For example, we have a nickel NTA chip in which we can capture a histag protein. We have a streptavidin chip um, in which we capture biotinylated protein, like that. So these are two different methods: a direct and a capture method. It depends on like what study we are like going to do. It depends on so this will help. I mean, what study we are going to do, that will governs like what immobilization process we should be like following. Uh, So next is in surface preparation. So this is typically how the I will show you about the one of the chip that is called as CM5 chip. So this is typically how the chip looks. There is a glass support or a plastic support, a gold layer, and a dextran matrix adhered to it. So why adhere to it? To this dextran matrix, the derivatives are attached, which helps us for immobilization. For example, in CM5 chip, carboxymethylase groups are attached, or any CM series chip, that is carboxymethylase series chip, the carboxymethylase groups is attached. This carboxymethylase group is explored to bind, is activated to bind to the amine group of our ligand for immobilization. So also why gold layer? Because it provides a metal plasmons, and it is inert in nature. And why dextran? Because it is flexible, it is less non-specific, high binding capacity, it withstand extensive, I mean, it is more stable. That's why dextran matrix is used. And also dextran matrix is like a, it is present in like a thread. So when we put the chip in this and when the buffer is flown, these threads can move. So it can nearly give us give it can nearly give like nearly give a phenomenon like a solution. So you can like it looks, I mean, it resembles like your protein and protein are interacting in a solution. So there are, so that's why it is given as a thread like structure. So now there are different types of chips available. It depends on like what experiment we are going to perform. So the commonly used chip is CM5 chip, that is, and other CM series, that is CM4, CM3, C1. SA chip is streptavidin chip, NTA, L1, HPA. L1 and HPA are used
You're
to
still on
immobilize
slide
liposome.
10, ma'am.
Huh? Hello.
You're on slide 10.
Oh, is it better like this?
Yes, ma'am. Better like this.
Okay, so I think there is some problem when I'm putting it in a like full screen. Now is my screen got changed? I mean, is it visible
Yes.
12th slide? Okay, so should I carry like this?
Yeah.
Yeah, okay. So, okay, so we were discussing there are like different types of chips available. It depends on like what experiment we are going to perform. We use this kind of chip. For example, if you want to capture your histag protein, you will going to use NTA chip. If you want to capture biotinylated biomolecule, then SA chip. So it depends on like what experiment we are going to perform. We use the chip. The most versatile chip is CM5 chip, which is commonly used. So these are different chips available, and these are the some information like what chip used when. So in CM5 is the most versatile chip. So if you're finding, if you want to uh, do low immobilization, then you will go for CM3 chip. For cells particularly, we use C1 chip. For whole cell particularly, we use C1 chip, and NTA for histag and SA for biotinylated, and L1 and HPA for capturing lipids and liposomes. So now what should we consider while immobilization? So the most foremost, the important thing is immobilization level. What should be the immobilization level? For example, when we are doing an experiment for specificity, so we can go for like wide range of immobilization. For concentration analysis, we will use high immobilization level. For affinity, it is low to mid, and for low molecular weight, low molecular weight for small molecules, we are going to use mid to high level of immobilization. Then what is the weight of interactants? Like typically, we immobilize higher molecular weight on chip, and we flow the lower molecular weight. So all these informations are needed, and what are the functional group available for binding. Also, if for example, if you're going to use amine coupling, so you need to be sure that amine group, whatever amine groups are available in the chip, in your protein, should, I mean, if you're studying a protein interaction, and if you are immobilizing one of the interacting partner by using amine group, by using amine coupling, and if that amines are present in a, like, present in a active site, then you should go for a capture method. So these are some considerations you should be taking care when you are doing the immobilization. Now, next step is, the first step is immobilization. Second is sample injection. In this, analyte is flown on the immobilized ligand. So in this, we need to optimize, like, what should be the flow rate, what should be the binding time, dissociation time. All these things we need to optimize. So this can be done by trial and error only. So there is one another point which is called as reference surfaces. That means our chip has 4 flow cells. That means 4 grooves. So our chip has 4 surfaces. That means 4 grooves, in which we can use 2 at for one experiment. So one is a test reference, and another is a reference. So first, the one is a test and another is a reference. So we immobilize our ligand on test, and reference is kept as is kept ideal. Now the analyte is flown from reference as well as test both. And for example, in our experiment, if you are using flow cell 1 and 2, so 1 will be kept as a reference, and flow cell 2, where we will immobilize our ligand. And analyte is flown from 1 and 2 both. And the data what we will get is 2 subtracted by 1. That means we are uh, subtracting the base. So if we subtracting the reference, if something is binding non-specific to the chip, we need to subtract that. Other subtraction is buffer subtraction. We, along with our analyte, we will also flow only buffer like analyte, and that is also subtracted. So this is called as double referencing, in which we subtract a buffer uh, and a buffer and the reference. That means whatever data we get here is the only interaction. For example, this is a binding happens at reference surface, and this binding is at active surface. That is called the test surface. So this active surface is subtracted by reference subtract, and this is how we get the final binding. So we are sure that for example, if it is a protein-protein interaction, here we are sure that protein A and B both, I mean, this interaction is only between the protein. There is no other effect for of buffer or a reference. So this is how typically it is shown. This is active, and this is reference, and this is corrected one. So what are the sample considerations? The sample should be homogeneous, it should be of good quality, it should be active, it should not get aggregate, and we have to take care of non-specific binding, and the injection time and all, we need to optimize that. And buffer should be compatible with the interacting partner. It should be filtered, and if it is giving us non-specific binding, then P20 is a surfactant which this can be added. So, and we also need to check whether our analytes are dissolved in any organic solvent. For example, in case of small molecules, generally it is dissolved in DMSO, so we need to correct that. uh, we need to do the correction of that. No, so these are the considerations we need to take care when we are passing the analyte. Now the first step was immobilization, and then after immobilization of ligand, we have passed the analyte, that is the binding step, and the third step is regeneration. So in regeneration, so as we were discussing, like um, when so when in the first step, the ligand is immobilized on the chip, and then analyte is flown, and then buffer is flown, so that the analyte will come off. As the analyte is flown, we get the association, and when the buffer is flown, we get the dissociation phase. Now, if the analyte is not coming off completely, we need to remove the analyte forcefully. That process is called as regeneration, so that we can pass another analyte concentration or another analyte over the ligand. So in regeneration, we need to be sure that analyte is coming off completely without affecting the ligand or its activity, like that. So we can check it like this. For example, this is a baseline, this is the association, and this is dissociation, and this is after regeneration. So we need to check whether these baselines are matching after regeneration, so that our another set of analyte can go, and it should give us the similar, I mean another set of analyte should go. So this is how we need to take care. So what should be the considerations? Like, it should not be harsh, or it should not be so mild that it should not be harsh that it is affecting the ligand, or it should not be that mild that it is not the analyte is not coming off completely. So what are the different um, degeneration buffers we can use? So there we can divide it into three: strong, intermediate, and weak, depending on upon the what interaction we are getting. We can use, I mean, we can use any buffer. So we need to do trial and error for that. So this is another type of another type of optimization for this. So the what are the in a summary, we can say that there are three steps in assay development. First is immobilization. Second is analyte binding, where association and dissociation is happening. Third is regeneration. Now, just to give an example, I would like to discuss one of the standard protein, that is protein and antibody interaction. The antibody is beta 2 microglobin, and uh, and uh, um, and the protein is beta 2 microglobin. Um, so here in this standard assay, we use chip, which is a CM5 chip, and a amine coupling is done for immobilization. As we have discussed earlier, the first step is immobilization. So in immobilization, we are going to immobilize B2M antibody, that is beta 2 microglobin antibody, and we are going to use amine coupling for this. As it is shown in this setup, that this is how we choose the chip. What are the different chips we can use? Here we are choosing a CM5 chip, which is more versatile. And there are 4 flow cells, out of which we are using 2 flow cells. One is kept as a reference, and another is a test where we immobilize. Now, after putting this is the typical like amine coupling method, in which carboxymethylase group is activated by EDC, passing EDC and NHS, and then and then a ligand is bound, and the unbound activated carboxymethylase groups are deactivated by passing ethanol amine. So this is for flow cell 1, and this is for flow cell 2. In flow cell 1, ligand is not passed, only it is activated and deactivated. We call this as a blank immobilization. And in flow cell 2, we activate it. We activate the flow cell 2, that is, we activate the carboxymethylase group, and we do the amine coupling. The amine group of our protein will bind to the carboxymethylase group, and it will attach covalently to the flow cell 2. Now, so this is how typically the result looks. So this is activation of the surface. Now we will pass the ligand, and then it is deactivation of the unbound. After binding of ligand, there will be few unbound carboxymethylase group which will be deactivated by ethanolamine, and we can see there is a difference between the baseline and the immobilized. So this is the level of immobilization we have achieved. So after immobilization, we are going to set up a kinetic assay. in kinetic assay, uh, so the contact time, the flow rate, and the dissociation time, we need to optimize it for every time for your new experiment. You will need to optimize this. Also, we will need to optimize the regeneration condition and the contact time and flow rate, everything. So these are the optimizations you will be doing every time. So this, and after that, we will be passing different concentrations of analyte. Generally, different concentration of analyte from low to high concentration. And then after setting up this, we get sensogram like this. This is association and this is dissociation. And these are the information. We get KD, KA, and K off. We'll discuss it in the next slide. So we have got concentration dependent binding, and here we can see the kinetics. Now, uh, like we uh, concentration dependent binding, and here we get the kinetics. So now, like, till now what we know is, like, what information we get is about our assay is the binding and the strength. So binding will tell you, like, whether your ligand and analyte are binding or not. Strength will tell you the how strong is their binding. So if we, I mean, this information can be derived from many other uh, techniques. So why SPR? So SPR is a very SPR is a very uh, important technique, because it will not only tell you KD, it will also give you information about the K on and a K off. So KD is equilibrium dissociation constant, and Ka and small k and small kd is a rate of dissociation constant and rate of association constant. So this information is very important, especially, for example, if you can see this theoretical sensogram, in which you can see that all of them will give you a same kd. You can see, like, those 4 5 sensogram, they are giving a same kd. That means their strength is similar. Then what further information we are deriving from the sensogram is, we are getting different Ka and different Kd. That means there is a difference in a on rate and the off rate. So this information is very important, especially if we are designing a drug molecule or biosimilar studies like that. So even if you see the sensogram, this will show you. So this is the association phase, and this is the dissociation phase. If you can check this green and the blue sensogram. So even if they are giving us same strength, there is a huge difference in a pattern of the sensogram. If you can see there, here we can see that it is very fast binding and dissociation is very slow. Here the binding is optimum and the dissociation is also, like, it's an optimum. In this teal sensogram, it is completely dissociating. But so hence we can know that the rate of association and dissociation are different. But if since kd is small kd by ka, the ratio can be same. So we in so for example, when we are designing a small when we are designing a drug molecule, for example, you are having 10 active compounds. Out of them, 5 are giving you same strength. Then how will you go further? So for that, this information is very important, the rate of association and the rate of dissociation. For example, if you are studying a sleeping pill, in that case, you would want to have an optimum association and an optimum dissociation. You would not want that it is binding and it is not coming off like that. So, or it is coming off very late, or it is coming off very fast. So this information is very important. And if you are studying a biosimilar, so in that case also, biosimilars, this information are very crucial, the rate of association and the rate of dissociation. I'll show you so how different sensograms look. So um, how different sensograms are, like, they look. So these are association and this is dissociation. You can see there is a difference in the shape of the sensogram. So when we are studying biomolecular interaction, it is important to know the strength, that is Kd. Also, it is important to know the shape of sensogram. That means it is important to know the K on and K off rate. So this information is very helpful. So these are rate constant. Ka, small k, and small kd. So small k will give you information about the formation of a complex, and kd is a dissociation. These are the units, and rate of this small k is dependent on concentration, and kd is a concentration independent. Now, after this, what are the applications? So, what specific key questions it addresses? It gives a specificity. It gives you information. Does the interaction is happening or not? The first thing is we would want to know the binding. First of all, how specific is the binding? Then we will want to know the kinetics. How fast and slow is the kinetics? The one which we are discussing, like k on and k off. Then affinity, what is the kd. This information will further help us in studying biosimilars, immunogenicity, etc. So the major application is, we know, like the kd and the k and k off will give you information of fundamental informations about the I mean, about the biological system, and how, and this k on and k off rate will tell you that how it is regulated, like what is binding to what and how this binding is regulated. This information can further be explored to different drug discoveries, and to check the efficacy, biosimilars, ligand fishing, etc. We can also develop a biosensors. I mean, if you want to particularly check any binders, so we can also design a biosensor. So these are the basic applications. So I'll give you a few examples what we have done it in our facility. So this is a protein drug compound. So these are active drug compound, and we see, so protein was immobilized and compounds were flown, and we have got the information about the KD, K, and K off. Now the protein and the Fab fragments. So we have done, I mean, the screening can be done, and then a liposome and a protein interaction, in which a liposome was immobilized on HPA chip and protein was flown on, and we have got the concentration dependent binding, and the KD. DNA and the protein. The protein was immobilized and DNA was flown. Also, we can do a temperature dependent. So in SPR, the particularly what we have it here is in Biaco T200, the temperature can range from 4 degrees to 40 degrees Celsius. So in that, we can study interaction kinetics, variation, whether it is temperature dependent or not. So this kind of studies can also be done. So this is just a uh, pictorial slide in which we can do a ligand fishing. For example, if we know the specific target, we can immobilize a specific target, and from the crude sample, we can we can bin it. We can have the specific binding, and we can elute this binding, and then we can check it in a MALDI or mass spec, and we would know that we can do the ligand fishing, the immunogenicity testing. For example, anti-drug antibodies can be checked here. Um, Another, we have already discussed about the drug discovery. Like if we are going to, I mean, this k on and k off rate are very crucial in this. So, for example, we have already discussed that if the KD is similar, the further k on and k off rate will give you like more information, like the rate of association and the rate of dissociation. So you can, like, we can do further detailed study by using this information. Other is, we can compare the sensograms. So this can be used in biosimilar testing, batch to batch variation, or if there is any mutants. So we can check all this. We can check all this since we can compare the sensogram. Now, this is the study we have done in our facility, that is antibody and the whole cell. So antibody was immobilized and whole cell was passed, and we want to study, there was a protein which was expressed on a cells, and we have passed whole cells, and we have seen the concentration dependent binding. It was only a binding study. So this is all applications we can be done by exploring the protein-protein by exploring the biomolecular interaction using SPR. So in conclusion, type of assays, binding, we can, like, the basic is binding. We can know yes or no, whether it is binder or not. The specificity, for example, what we were seeing in, like, in ligand fishing and all, the specificity, the kinetic, the strength of a binding, and the K on and K off. And the concentration analysis can also be done. So we can this kind of assays can be done for wide range of biomolecular interactions, from protein, from nucleic acid, lipids, carbohydrate, small molecule, bole cell, virus, bacteria, etc. Like, we can also run the lysate crude sample, and we can see we can see the binding or the kinetics there. Now, in conclusion, SPR is an optical technique which is used to study biomolecular interaction. It is label-free technique, and it is, we can study interaction in real-time manner. So you can we can go for for immobilization, we can go for capture method as well as direct method. Three major steps. First is immobilization, in which we immobilize one of the interacting partner, that is called as ligand, and the other partner which we flow is called as analyte. Second step is sample injection, in which we consider, we have to optimize, like, what concentration of the sample, and what should be the flow rate, what should be the time, what should be the dissociation time. After all this optimization, we will pass the sample. Also, we need to check the regeneration conditions, and we need to also consider what buffer we are going to use um, for our sample. And we need to do referencing also. That is, the chip has four flow cells, in which two flow cells are used for one experiment, and the first flow cell is always kept as a reference, and second is a test. And the data we'll get from test minus reference. And we will also run a buffer as an analyte, and buffer is also subtracted. This is called as double referencing. And applications are major applications are binding, kinetics, and concentration analysis. The sensogram, and it is going to give you information about the strength of binding, also the KD and a KA. And this rate of on rate and off rate is very important to study. Also, this sensogram shape determines the kinetics. Uh, we need to consider this, and this KD is K off by K on. So this K off rate and on rate will be very informative, especially when we are designing a drug molecule or biosimilars or etc. Thank you. Any questions?